Hi students, welcome to week number 2 of your Biology 120 lab. Let's take a look at what we did last week and then we'll see what we've got going on this week. Okay, so last week we examined the scientific method and I talked about it as a process where scientists examine phenomena that happen in the world. We design experiments, we carry out those experiments, analyze the data, and publish our work. And this is how science is done. I told you how important the peer review process was because it is tremendously important. It keeps everybody honest. You also did some graphing. You practiced graphing by hand and you took a look at those wheat and rye plants and how they had grown over time. But you also used Excel, if you have Excel or a program similar to that, to graph by the computer. Now, one thing that I don't want you to do is forget how to do this because you're going to be graphing more through these labs. So be aware of that and realize that you can always go back to the instructions in this lab number 1 to help you with using Excel and to producing graphs, making linear regression lines and that sort of thing, and finding those equations. Okay, so what do we have going on this week? This week is all about the chemistry of life, macromolecules, pH, buffers, the importance of water to life, all those sorts of things. And sometimes students wonder, well, why in the world do we have to study chemistry? This is a biology class. Yes, but all of life is based on the chemistry of life. And so we do have to start out with common terminology and common ideas and understandings about chemistry. So your experiments this week are on pH and buffers. Well, what is pH? pH is a measure of how acidic or basic an aqueous or water solution is. So what are you measuring? You're measuring hydrogen ions, which is represented by capital H plus, and that tells you it's a hydrogen ion missing an electron, or hydroxide ions, which is represented by OH negative, and that's what's left over from a water molecule after that hydrogen ion splits away. So if you have more hydrogen ions, the H's, it's an acidic solution. If you have more OH ions or hydroxide ions, you have a basic solution. So why is that important? Because most of life wants to be held at a neutral pH. You don't want too many ions that have positive charges or negative charges in your cells because that messes up all of your molecules. So most of life wants to stick to a very neutral pH. And if you move on either end of that pH scale, you're going to do some harm to organisms. We'll also take a look at buffers. A buffer is a chemical system. It's usually 2 different chemicals that are in a solution that can manage pH or hold it constant over a certain range. You know, you can't overwhelm a buffer and then the pH will change. But buffers are there to maintain pH in solutions. We have buffering systems in our blood. So that's just one example, but there are plenty of buffers out there. We use buffers all the time in molecular biology to keep pH constant. Okay, what else do you need to do this week? Review the introduction to the chemistry of life and the overviews for Module 2. Realize, I didn't realize this, but realize in the introduction, this is where you have the lab instructions. And I did make an announcement where you and show you where you can find those. Your lab instructions are in the introduction. Read through your lab instructions. Gather all your supplies. Your supplies are in the kit except for water, I think. Labeling will be very important for the little tray that you use. You need to label it correctly, and you're going to show me that in some pictures. Labeling is terribly important because otherwise, how are you going to know what you put in each little well of the tray? So labeling is terribly important. Perform the experiment on pH, and then there's one on buffering, and record the results appropriately in the lab. It's really important to mix your liquids before you come to a conclusion about the colors that you see. So make sure you do that, and you can either do that by swirling around the tray a little bit, be very careful with that, or gently pipette the liquid up and down several times. Uh-oh, I'm going to sneeze. Nope. Okay. So gently pipette up and down, or gently swirl your tray to get your solutions mixed up so then you can read the colors. And you're going to take a couple of photographs to include in your lab report. Okay, then you write up your lab report. The lab report form is in this lab. It's actually under course menu, start here, assignments and guidelines. I give you those instructions in the announcements too, so that you write up your lab report correctly, and you will include your photographs. Now, one word. All of these reports that you're turning in need to be a single Word document. So don't turn in the lab report and then 2 separate photos. They need to be in the document itself. Okay? This is due by Sunday evening by midnight, preferably before midnight, so there's no question. So that's what's going to be due this week. I'm going to put in the announcements, this little YouTube video, which is very good. It's only 3.5 minutes long, on what is pH and what are buffers. So realize that you have that. And then also this week, you're going to be looking at your final project details. So you will be doing a final project like you are in lecture if you're taking lecture this term. It will be different though, because it will be experimentation that you're going to choose to do and modify. And then write up those results. So what you need to do is choose one experiment. Look under the final project explanation section for week number 2, module number 2, and you click on that, and it will have a link there for Ask a Biologist. Ask a Biologist is a website, and there are 4 different experiments that are chosen for you. The ice one, the nerve one, I've forgotten, the caterpillar one, and there's another one. I forgot now. Those are chosen for you that you can possibly do. There are a couple of others. There's one about breaking proteins. That's a good one. And the virtual pocket seeds, that's also a good one in that list from Ask a Biologist. Some of them are not really good. And not for you to do, you know. Because some of them are just simulations. So take a look at those and decide on which one you want to do. If you want to do one of those, and of course realize that you're going to do it, but you're also going to modify it in some way. You're going to be looking at a bigger picture than the one that they're talking about in this experiment. If you want to do something totally different, you have to get this approved by me. And really, you need to do that this week. So if you're thinking of doing something else, you need to ask me about it, including any of the other experiments that are on this website, Ask a Biologist. You need to get this approved by me so that I can take a look at it and see if it is doable. Sometimes people come up with fantastic ideas about research that they want to do, but they can't get it done in 8 weeks' time, really 7, because this final project is due the 7th week. So what you're choosing to do has to be doable in our time for this term, and I can help you figure that out. So if you have any questions, as always with me, I'm just an email message away. Please send me an email message. I also hold online office hours on Sunday evenings starting at 6 o'clock. I started a little bit early last night and finished a little bit early because WebEx kicked me out. But I'll start this coming week at 6 o'clock. Feel free to stop in and ask me questions, anything that you need to know, or email me, or post under the general questions in the discussion area. I answer those too. If I were you, I would subscribe to the general questions discussion so that you get notifications about that, because I will be posting some things there too. So I hope you have a wonderful week. It's after the holiday now, so we can really get back to work and get going. You can start looking at that final project, figuring out what you want to do, and work on pH and buffers this week. I hope you have a wonderful week. Let me know if you have any trouble or any issues.